Hey Mustangs, in this video we're going to be taking a look at cancer and the second category of genes that are involved with cancer called tumor suppressor genes. Now in the previous video we talked about proto-oncogenes and I'm going to refresh your memory a little bit about that in this video here. So first of all, a bit of a reminder, cancer is one of your cells dividing out of control. So when one of your cells gets mutations, damage to its DNA, in certain types of genes, it's going to lead to that cell just dividing out of control. The cell cycle, which is involved with cell division, is very specific and it's regulated to where you have certain signals that tell the cell to continue and certain signals that tell the cell to stop. But in a cancer cell, the DNA that controls those signals, whether to continue or to start or to stop the cell cycle, they've been damaged. And if they've been damaged, the cell could start to divide out of control. So the two classes of genes that can affect whether a cell becomes cancerous or not. The first we talked about in the previous video, the proto-oncogenes. So the proto-oncogenes, these are going to be genes that code for proteins that are signals that tell the cell to actually divide. So they help the cell move through the cell cycle, telling the cell that when it gets to a checkpoint, everything's good, you can move on to the next step. If proto-oncogenes become damaged and they start making these signals when they're not supposed to, those signals are basically going to tell the cell to divide. And that's what cancer is, cells dividing out of control. So these signals are turned on when they're not supposed to. So whenever a proto-oncogene becomes active, we call it an oncogene instead. And then the second category is what we're going to take a look at the rest of this video, which is the tumor suppressor genes. Okay. So with tumor suppressor genes, these are the opposite of the proto-oncogenes. The proto-oncogenes made signals that told the cell to move on and continue and go through the cell cycle to divide. Tumor suppressor genes are going to code for proteins that are going to tell the cell cycle to stop. So it's going to tell the cell to stop going through the cell cycle. Okay. Many of these genes are going to stop cells at checkpoints. They can be involved in repair of DNA or even in activating the proteins needed for apoptosis for the cell to die if it's too damaged. Now for the proto-oncogenes, we've identified about 40 in humans. For the tumor suppressor genes, we've only identified about 10. So there's only been about 10 tumor suppressor genes identified. So out of the approximately 19,000 different genes that we have, about 10 of those are tumor suppressor genes. They make proteins that are signals that tell the cell, stop, don't go any further, there's something wrong, we need to fix things. Now, the example that we're going to be taking a look at is going to be the p53 gene. So, the p53 gene is found on chromosome 17. So on the short arm of chromosome 17, you're going to find this gene. The gene makes p53 protein. Those p53 proteins have three major jobs. Now remember, this is a tumor suppressor gene. So the first major task or job that it has is it's to tell the cell to stop at the G1 to S checkpoint. Okay? So the p53 proteins that are made from the p53 gene will tell the cell don't go past the G1 to S checkpoint. The p53 proteins also help with repair of DNA, and they also help with apoptosis. So we're going to break these down and look at 1, 2, and 3 individually. So let's start with number 1. When DNA gets damaged, it's going to cause p53 proteins to become phosphorylated. Now whenever a protein or a molecule gets phosphorylated, that usually means it's getting activated. So phosphorylation equals activation. When the p53 becomes phosphorylated, when it becomes active, the proteins, basically the p53 is basically a transcription factor. If you think about what a transcription factor is, it's going to be needed to start the transcription of DNA to mRNA, and then once the mRNA has been made, it's going to be translated into a protein. So p53 is a transcription factor that's going to bind to the promoter of a certain gene called the WAF1 gene. That WAF1 gene makes p21 proteins. So a lot of information here, but we're going to break it down with a diagram in a sec. The P21 proteins, here's why it's important for the P53 transcription factor to activate this gene here. The P21 proteins are a signal that stop the cell at the G1 to S phase checkpoint. So the cell goes into arrest, it stops, and the way the P21 does that, it binds to certain cyclin CDK complexes. Best way to think of P21 is as an inhibitor, because CDKs, remember, they are enzymes. So whenever you have a molecule that will bind to an enzyme and stop it from working, you're looking at an inhibitor. So the P21 proteins that we see here, they bind to the cyclin CDK complexes and they inhibit them from being able to do their job. Think about what cyclin CDK complexes are. These are going to be what are signals that tell the cell to move past the checkpoint. If the P21 binds to this complex, the signal is turned off. It can't tell the cell to move past the G1 to S checkpoint. So P21 turns off that signal and basically stops the cell cycle. So let's go ahead and take a look at that with a diagram here. So it all starts with damaged DNA. So if the DNA becomes damaged, so I'm going to show this as damaged. Actually, let me pick a different color, let's say. So the DNA has become damaged. So we'll start with damaged DNA. That damaged DNA starts a phosphorylation cascade. So you have a relay molecule, usually it becomes phosphorylated, that means it's active. Then it will activate the next molecule by phosphorylating it, and that activates the next molecule. In this case, it's going to be P53. So I'm going to draw P53 here. P53. This molecule will phosphorylate the P53. Now, just like we saw in the previous slide, whenever P53 becomes phosphorylated, it is now turned on. So this is now a transcription factor that's going to head into the nucleus. So it's going to go into the nucleus here, and it's going to bind to the promoter region of the WAF gene. Now, I'm not going to draw the double helix. I'm just going to draw a single part of the DNA here. But let's say this is the WAF, part of the WAF gene here, and this is the promoter region. It's going to bind to this region here. So P53 is going to bind. Once it binds, so here's my P53, it binds to the promoter region. That's going to cause other transcription factors that are necessary to start the transcription of this gene to bind as well. Once you have all of the transcription factors assembled along with the RNA polymerase, now you're going to transcribe the gene. Okay, and this was the WAF1 promoter. It's not working for me here. Okay. Once you've transcribed, remember when you transcribe, that means you're turning the DNA instructions into mRNA. So we're going to say this is a P21 mRNA. Once the mRNA has been made, then translation has to occur to make a protein. When the protein is made, when translation occurs, it makes the P21 protein. So I'm going to make the P21 protein here. So P21. So this P21 protein, that's what we want to make. When this P21 protein is made, it's going to be able to be an inhibitor to the cyclin CDK complexes that would normally tell the cell cycle to continue on. So over here, let's take a look at what happens. So here's the cyclin, and it's bound to a CDK. When they're bound, remember that's active. If they're separate, that means they're inactive. So active, when these are bound together, that means it's going to tell the cell, so tells the cell to move to, in this case, we want to go from the G1 to S, so this is going to tell the cell to move to the S phase. Okay? And that's if these are active. So what happens when P21 has been made Here's my cyclin CDK complex once again. Sorry, my pen's just not working right now. So cyclin CDK complex, but this time the P21 is going to bind to it. So here's my P21 protein. It binds to the complex, it deactivates it. So now it's inactive. It's inhibitor. It's an inhibitor. So it's inhibiting the cyclin CDK complex. It's inactive. That is going to tell the cell to stay. So cell will stay in G1. And again, the only reason why the P21 was made to inhibit the cyclin CDK complex is because the DNA was damaged. We don't want the cell to continue on to the S phase and make copies of the DNA that's damaged. That's going to be a really bad thing. Okay? So this is a way of regulating the cell cycle. So it stops the cell cycle in G1. Once the DNA has been repaired, then it'll move on to the next step. Okay? So P21 is essential in stopping the cell cycle if the DNA has been damaged. So remember the three major tasks again of P53. You have stopping the cell cycle at G1 to S, you have helping repair the DNA, and then you have helping with apoptosis. So just a quick little breeze over of the other two. So when it comes to repairing DNA, the P53 proteins are going to help activate the DNA repair genes. So P53 can activate certain genes that are going to help repair the DNA. And for number 3 here, help with apoptosis, P53 can help activate the genes that are going to be enzymes used to kill the cell. So remember, apoptosis is killing the cell, where there's such a big problem that can't be fixed, so the cell has to be destroyed because it doesn't make sense to make copies of it. It's too damaged. Okay? Now, all of this that we see here is all about regulating the cell. So if the P53 gene is damaged, the cell can no longer stop and repair DNA. And if you think about it, if one of the parts of the DNA that's damaged is a proto-oncogene that is telling the cell to divide, P53 would stop and repair it to make sure things are good. But if P53 is mutated and it no longer functions, it can't stop and repair that DNA. And because of that, you have cancer.